Thank you, Jessica. That was a fantastic talk and discussion. I really learned a lot and have a lot of questions for you myself, too. I'd like to invite my co-chair and colleague, Nabeel Bardeesy, to give the next talk. As Stacy mentioned, Nabeel and I met through the Cholangiocarcinoma Foundation maybe in 2012 when we had just started a tumor bank at UCSF of our patient samples coming from the operating room or from biopsies or leftover from surgeries long ago, with support from the Billy Project Foundation. I don't know if Sue Acquisto is here yet, but we had that growing bank of samples and data, and then Stacy saw that resource that we were building and knew that Nabeel's lab was doing studies in mouse models and cell lines and could use our samples from UCSF if we could get them to him in our data, and connected us and actually gave us some grant funding to start the process of sending Nabeel's lab samples. And since then, I've had the great privilege and awe of watching his lab do great work with so many different molecular subgroups of cholangiocarcinoma, and I'm very privileged to get to work with him and introduce him today.
Thanks so much, Katie. It's a great privilege to be presenting here today to you all, as well as to be affiliated with the foundation for all these years. And finally, it's been great fun working with Katie, of course. My laboratory studies cancer from the laboratory perspective. And I have to say that when we started to study cholangiocarcinoma in around 2009, it really was not a large research area. So one way you can gauge this is the number of federally funded grants on cholangiocarcinoma. It was vanishingly small. Encouragingly, this is really starting to change very rapidly. And in fact, the choices I made for what to present today sort of inform part of this increase in research that's happening. So first of all, I'm going to speak about the genomics of cholangiocarcinoma. The genomics, and I'm going to define that broadly, has allowed clinicians to have new hypotheses to treat cholangiocarcinoma patients, such as the FGFR mutant group of tumors. And also this genomic information has brought people who study these pathways that are altered in cholangiocarcinoma. It has made them curious about how the pathways they study are manifest in these other tumor types. And so that's one of the framing points that I've used, Katie and I used to invite speakers today, is to the basic science portion of the meeting starting tomorrow, which is trying to bring in experts who are really in the top of their field in relevant biological pathways to cholangiocarcinoma, but who might not have invested their effort in that area. So in one hand, it was to expand the discussion and hopefully to further attract people to the field. The second framing point that I'm going to go over briefly, and Katie alluded to it, was model development. So in one of the types of models the field uses is cell lines. So this is material derived from tumors that can be grown in the laboratory. And in cholangiocarcinoma, until very recently, there have been very few of these models, and what were available were primarily from East Asia, where the risk factors are different, as you heard from the beautiful talk by Jessica. So one of the things that having a active effort to develop models enables is to study disease subtypes, but also, again, to have systems that many, many more people can enter a field. And so I think some of these themes are going to recur in the meeting, is this collaboration, bringing more people into the field, and being able to study the disease in all its complexity. So from there, I will provide an overview. Okay, yes. So I'm going to provide an overview for cancer genomics. And again, as I said, I'll define that broadly. And I'll also try to explain some of the underlying principles, and I hope that interests you. Of course, the ultimate reason that we're doing this is to predict subtypes of cholangiocarcinoma, to understand how the disease is different between individuals. Also, very importantly, to predict new therapeutic approaches. And that's actually led to a lot of very interesting clinical hypothesis that's coming to fruition. But illustrating both the challenges, the opportunities and challenges is, taken as one example, the tumors, cholangiocarcinomas that have FGFR mutations. So I'm going to speak a little bit about the state of the science in that area and the challenges and also opportunities that are in that area. So broadly, what are we trying to do with genomics? We're trying to understand whether there are differences, for example, gene mutation-wise, between cholangiocarcinoma occurring inside and outside the liver, for example. Until very recently, these had been treated almost identically in clinical trials with chemotherapy. But we're starting to learn that, in fact, these diseases are quite different and need to be resolved further into subtypes. In addition, within even an anatomical subtype, we're learning that the disease can be manifested very, very differently between individuals. And genomics is helping us understand those subtypes. And then using this information, can we define much more specific therapeutic approaches for individuals once we discern meaningful subtypes. And so to further resolve this, the types of questions that the field is asking and have started to address very effectively are what genetic abnormalities, so either mutations in genes or loss or gain or loss of chromosomes. And the second related area is what genes are turned on or off in cancer, and I'll get into that in some detail, and why we want to know those questions. So apologies to people who probably are quite familiar with this, but I'm going to go very briefly into some of the underlying principles. And again, I will not belabor these points, but of course, cancer is a genetic disease, and we have 46 chromosomes that encode the genes that conduct the functions in the cell. If we look a little bit closer, each of the chromosomes, each of the 46 chromosomes harbors a number of genes. There are 20,000 genes in the cell. And there's about 500 genes that the field commonly agrees can be abnormal in cancer, and changes in that gene specifically can contribute to cancer formation. So it's about 500 or so, but there's probably more than that. And within any individual cancer type, we can identify probably about 40 or 50 genes that there's good evidence that the abnormalities in those genes contribute to either the development of cancer or the ongoing growth of cancer as well. And of course, the genes that contribute to the ongoing growth of cancer might represent opportunities for therapeutic targeting. In addition, not only are genes mutated, so the gene has a change in one of the base pairs that makes up the sequence of that gene, but also the genes must be turned on or off. And this diagram is perhaps not the most pleasant to look at, but I got this on an NIH website for a kind of a general view that genes are controlled by machinery that determines whether they are actually activated in a cell or not. And I'm going to give a specific example of this. And the genes, when they're activated, are transformed into a material called RNA. And the RNA is the messenger that tells the cell which proteins to generate. So each of our cells in our body and in cancer cells have different complements of genes being on or off, and different complements of the proteins that are engaging those functions. And so what the field has been gradually doing is first of all, asking what mutations are present in cancer cells, and secondly, what genes are on or off, and at what levels are they on or off, and third, what proteins are present in the cell. And so therefore, we can identify subtypes of cancer with different sets of alterations that will hopefully predict therapeutic approaches. Okay. So I want to give one or two examples of how the identity of the cell is dictated by what genes are on or off. So some genes, this is a tubulin, this is a gene that basically keeps the cell structured normally. It makes sort of fibers within the cell. And this is just showing, the graph is showing the relative expression across many tissues. And I didn't blow it up because it's just basically listing tissues from the brain to the intestine to the kidney. And you can see that the relative height of the bars, do I have a pointer on this? Yes. The relative height of the bars indicates how much of that gene is turned on in each of these cell types. And you can see across the board, this gene is just playing a structural role, and all tissues express comparable amounts. But then there are other very, very specialized genes like insulin. And you can see here that it's really completely absent in all tissues except the pancreatic islet, which produces insulin after we eat a meal. So the genes that are on or off determine the identity of the cell and the function of the cell. Now, what about in cancer? So this is not a beautiful graph here. It's depicted slightly differently, but in cancers often abnormally have genes turned on abnormally. So this is a gene called Lin28B, and it's very important in the very early embryo of keeping cells primitive so the embryo can grow before the tissues form specifically into specialized brain and kidney and all the other tissues. So the function of Lin28B in the normal development is to keep cells very primitive. And you can see that in the adult, actually there's no Lin28B expressed. If I showed you an embryo, you'd see high bars here. The only tissue it's on is in the testes, so the male germ cells. So it's not on in any of our mature tissues. But in cancers, this gene gets turned on quite robustly. Each of these dots represents an individual patient here with these different types of cancer. And you can just see across cancers, and in fact often across particularly cancers that are metastatic, you have this abnormal genes being turned on. So I'm showing you these examples to explain some of the underlying principles. We want to understand the genes that are functioning abnormally by having mutations, but also what genes are on or off in the cell that dictate the functions of the cancer cell. And then finally, the other thing that genomics does is, in addition to looking for mutations and the level of expression, genomics looks for deletions or gains of chromosomes. So if you have extra copies of a gene that's normal function is to regulate growth, the abnormally high levels of that gene that can be either dictated by turning it on or off, or extra copies could confer an abnormal function. And how does this happen? So the previous talk was about epidemiology, and certainly most studies indicate that there aren't clear risk factors for most people with most adult cancers. So most adult cancers seem to be associated in part by the inherent machinery not functioning perfectly between cells. So what the machinery that I'm referring to here is the machinery to copy the DNA as cells divide. So the chromosomes need to be duplicated. So here's a chromosome. It's duplicated as a cell divides, and then the individual chromosomes are segregated into each of the daughter cells. So anytime a cell divides, all of the DNA needs to be recopied. And so the DNA is 9 billion A's, G's, C's, and T's. And so during that copying of this sequence, there can be a single incorrectly introduced base. And that can be with the unfortunate circumstance sometimes in a gene whose normal function is to regulate growth. But other kinds of genomic events that can happen in this kind of complicated chromosomal segregation process include deletions, and that deletion, if it removes an area of the chromosome that encodes an important gene that prevents abnormal growth, and some of the genes you might have heard of include P53, BAP1. These are frequently mutated in cholangiocarcinoma. So these things can happen as a consequence of the imperfection of the machinery to copy and segregate chromosomes. Another event that's relevant to a lot of people with cholangiocarcinoma are these translocations, where chromosomes get broken and improperly fused together, again in this process of segregating the chromosomes during division. So about 20% of individuals with cholangiocarcinoma have fusions involving FGF receptor. And so current genomics is trying to fully understand the landscape of these mutations that happen between individuals. So where are we to date? So what about the first question? Until I would say about 4 or 5 years ago, there really wasn't a clear view of the distinction between intrahepatic, intrahepatic cholangiocarcinoma and forms of distal or extrahepatic cholangiocarcinoma. We knew a small number of mutations, and they were common to both types. So there was no reason to have a distinct treatment hypothesis because they looked similar clinically. Now we jump to 2018, and this is an incomplete picture, so this is just shown diagrammatically, but I think I tried to point out some of the important mutations. So, in fact, gallbladder also was lumped in with intra and extrahepatic cholangiocarcinoma. And I just highlighted some of the mutations, in this case, that are unique to intrahepatic cholangiocarcinoma. Mutations in IDH1 and 2, and in FGF receptor. These are vanishingly rare in gallbladder and in extrahepatic cholangiocarcinoma. And of course, what's important about these is that pharmaceutical companies have developed inhibitors to these genes. So if we understand the function and whether cancers really depend on these mutant genes for continued growth, there's an opportunity for real therapeutic advances. So this big picture view has given us the sense that these cancers need to be studied. Definitely we want to know what's common, but we also need to really resolve them into anatomically separate subtypes and think of them as probably a spectrum of different diseases with some relationships, but clearly different underlying genetics. And then this is just an example just shown more concretely here, is that this is just simply showing the frequency of mutations in the IDH1 and 2 genes across a number of gastrointestinal cancers, including gallbladder cancer and I should have extrahepatic, I don't see it here. In any case, effectively, within GI cancers, the rate is extremely low, other than intrahepatic cholangiocarcinoma, and that includes very rare in extrahepatic as well. Another issue is within an individual anatomical subtype, can we identify very evident cancer targets, cancer drug targets, I should say. And in cholangiocarcinoma, unlike another difficult cancer that I work on, pancreatic cancer, there are in fact a number of mutant genes for which there are immediate hypotheses for how to treat this with a very specific drug. And so that gives opportunities, again, for very specific therapeutic approaches. So one of them are these mutations in IDH1 and 2, and these are currently being treated in clinical trials with specific inhibitors, and these are showing promise. FGFR2 fusions, translocations, point mutations. As you've heard from Porter, these are showing really also an impressive amount of promise in those patients. In addition, BRAF mutations. There are inhibitors for BRAF that have shown efficacy particularly in melanoma, but a number of other cancer types. These mutations are also present in a small subset of cholangiocarcinoma patients. Finally, PIK3CA mutations. Again, this is a mutation for which there are a number of drugs that can overcome in the clinic. So this is a completely different landscape than was evident even 4 or 5 years ago. And again, back to one of my original points is that, for example, I invited to speak to this meeting a very top oncologist and cancer researcher who's an expert on IDH and who's been studying in glioma. And so by working together, we can understand what might be common about the vulnerabilities of these tumors in different compartments. And that's the true for a number of, I also invited somebody who works on NF pathway genes, again, who hasn't really thought of it in this context. So we can, despite cholangiocarcinoma being relatively rare, the genes that are mutated in other cancer types, and hopefully the collective wisdom of different fields can tackle the challenges of targeting these mutations. Now, we can go beyond looking at mutations to understand subtypes by using, and I apologize for this, I will explain this slide a little bit more. But another thing that we can study is the genes that are turned on or off in a cancer cell or in any cell. And so instead of looking at one or two individually, like I showed with insulin, there are computer and micro chip-based approaches for determining the levels of each of the 20,000 genes in a cell. So we can look at insulin, we can look at digestive enzymes, we can look at neurotransmitters, and genes that are involved in growth, like FGFR, and determine the relative amount of that gene that's expressed in individuals with cancer. And what you see here, this is what we call a heat map. So red means lots of that gene. On this direction, this is a list of 20,000 genes or so. And then if it's red, it means that those, these are also individuals with cholangiocarcinoma. So these are 40 patients here. And this is the level of a large number of genes. And so if it's red here, it means that those genes are abundant relative to a median amount. And what these data are showing is that on a kind of broad level, one can classify the whole set of genes expressed by individuals with cholangiocarcinoma into 3 broad clusters. And so right now, we don't have a lot of insight into whether we can use this information to devise new therapeutics, but there are a few hunches that we can devise. And again, this is trying to go beyond mutations, because not everybody who has these types of cancer has a mutation that is readily targetable. But perhaps by understanding the broad cellular circuits, there might be obvious hypotheses. So this is not ready for any kind of prime time therapeutics. We have to look in the laboratory. But one thing one of the sequencing efforts that I was part of defined was that if one sort of zones in on part of this map here, one can see a broad theme regarding genes that are very important in certain cellular functions. So the mitochondria is the sort of energy factory in the cell, but it's also the machinery that interconverts many nutrients. And so it allows the cell to build all its building blocks. And a subset of cholangiocarcinomas, in fact, those with IDH mutations, tended to have much, much higher abundance of genes that are involved in these mitochondrial functions. Another subset had much more abundant levels of genes that are involved in turning on or off genes. So this is another way we can use genomic information. We need to understand it better, but it's allowing us to zone in on potentially discrete subsets of cholangiocarcinoma and lead to new clinical hypotheses. Finally, in terms of thinking about genomics, I said that I wanted to define it broadly, is that there's a great deal of interest now in understanding how cancers can either co-opt the immune function and increase inflammatory processes that encourage cancer growth, or to thwart the normal ability of the immune system to eradicate the cancer as a foreign element. And so many groups have been thinking for the last actually maybe 15 years about cancers as being a real environment of many cell types. The tumor cells, shown here, are the cells that have the mutation. However, the tumor cell can either recruit other cells that can either be trying to fight it or that can actually have an interplay and nurture the cancer growth. And the other cell types include blood vessels that bring the nutrients to the tumor, as well as a whole host of immune cells that can both contribute positively and negatively to tumor growth. And so genomics can also give us a sense of whether different subsets of cholangiocarcinoma patients have different recruitment of immune cells. And this will lead to hypotheses for how to toggle immune system function or how to modulate immune system function. And this is a work in progress here. There's no punchline that I wanted to tell you, but just that this is something that's rapidly developing and will lead to the next generation of clinical hypotheses that we can test. And of course, we can study this in the laboratory. Now, I want to spend the last few minutes in just talking about one specific case, because I think that many individuals here are in clinical trials and have these alterations. So cholangiocarcinomas have an unusually high frequency of these alterations in the FGF receptor 2, or fibroblast growth factor receptor 2. Now, this protein, it resides on the surface of the cell, and part of it's sticking outside the cell, and it can respond to fibroblast growth factors, which are produced by other cells. And I'll go into that in a minute. And then they have a portion of the protein that's in the cell. So this fibroblast growth factor forms a dimer. So 2 copies of itself are needed to work together. And inside in the cell is how it relays signals for how the cell should respond to engagement of these growth factors. So during the normal development of the liver and many tissues, the fibroblast growth factor, in response to neighboring cells producing these growth factors, tells cells to grow or to divide or to form mature functions. Individuals with achondroplasia have abnormal activity of this. So in the bones, this signaling pathway is responsible for cells deciding whether to keep growing or to mature. In cholangiocarcinoma patients and in another number of other cancers, you lose this normal need for an external signal, these growth factors. So normally this is very tightly regulated. And so these intracellular growth responses that I referred to that involve many, many different cellular compartments are under this very tight regulation. This is off only until there are growth factors. However, in individuals who have fusions, which are the most common mechanism of activation or point mutations, there's no need for these growth factor ligands, and the signaling is always on. And so always the ability to engage different cellular growth pathways are operative. And this is an old slide. You're going to see much more updated ones. But this was very, very encouraging results, is that the majority of people, particularly people with individuals with fusions and cholangiocarcinoma, showed benefit from this drug. And so this was very encouraging. It's telling us that not only are these mutations frequently found in patients, and probably contribute to the initiation of the cancer. And we have evidence in genetically engineered mouse models that indeed FGF receptor mutations get the cancer started, but also once the cancer is growing, the cancer remains dependent on this pathway for ongoing growth. And this is a very important concept that makes it very vulnerable to drugs that target the FGF receptor pathway. And there are many, many that have been developed and many more that are ongoing. And so this is a very encouraging response. And again, much more updated data has reinforced that. Now, some patients have very sustained responses, and sometimes the responses are not as robust. And so many groups, and including our group at Mass General with clinicians, and you're going to hear some of them tomorrow, Lipika Goyal and Katie and others, have wanted to understand what is the nature of the response, and when tumors do stop responding, what is the basis for that? And so one of the ways this can be done is to look at the genetic features of the tumor before treatment and to monitor if patients' tumors do progress, what is the difference in terms of gene mutations or other of the genomic features I spoke about, immune system and so on. And secondly, as a surrogate for what is happening in the tumor, it turns out that tumor cells are shed into the blood all the time, and you can detect abnormalities that are present in the tumor by looking at tumor cells that are circulating in the blood. So without any need for any biopsy, and you can do this over time in a way that's much less difficult for a patient by monitoring DNA for the presence of mutations. So I'll just show you a tiny bit of that data here. So this is a patient who originally showed a modest response, but indeed clearly the tumor shrank on an early clinical trial. But what happened when the patient's tumor recurred was that the tumor had acquired a number of other mutations. And what was interesting here were that these mutations were all in the FGF receptor. So the patient was treated with a drug that can turn off the FGF receptor. But the tumor became resistant associated with a number of other mutations that were detected in the blood and that were probably present in different metastasis, and that were, I should say, present in different metastasis that were resistant. And we've developed a lot of understanding of these mutations. So they were not present or they were not evident before treatment, but when the tumor recurred, these mutations were associated with resistance. And we were able to show in the laboratory, and also by understanding the structure of the protein, FGFR, that all of the mutations either were the original drug from Novartis called BGJ-398, they were exactly where the drug bound. So now that the protein has a different structure and the drug couldn't bind, or in other parts of the protein that either affected the access of the drug into the protein or that increased the ability of the protein to carry out its biochemical function. And all of the mutations were completely understandable. So certainly that the glass is half full here is that we only understood why the resistance happened. But the good fortune is that other pharmaceutical companies have developed completely independent inhibitors of FGFR that bind to different parts of the protein. And so a number of these patients have subsequently been treated with a second inhibitor, for example, a compound from a company called Taiho that is entirely different, and that those mutations now were not resistant to that inhibitor. So on a first level, we understand part of the mechanisms of resistance and also in part how to overcome it. And so these are very important steps in the right direction. But really to go to the next level, we need to have experimental systems where we can really model this and we can completely understand the biology of FGFR mutant cholangiocarcinoma. On one type of model we've developed, and I'm not going to speak about that here, is we developed genetically engineered mice where in the normal liver, in the adult liver, we can cause that mutation, for example, FGFR translocations to occur, and we can understand from the earliest stages of this disease what are the abnormal events that happen and maybe whether we can prevent, but also we can understand the cancer as it arises in its normal context. The second type of model that's been very valuable is that I referred to earlier as the lack of cell lines. And in fact, no cell lines can you order from the major repositories that are maintained by nonprofits. None of them have this FGFR fusion. So how can you really get to the next level of hypothesis? So with collaborators like Katie, we've developed a large bank of these that we're trying to validate. The models involve two kinds of components, and I apologize if I'm going over a little bit, I'm almost done, is from resected specimens or from biopsies that patients generally generously donate to research and consent for their use. We can either put it into patient-derived xenografts, which you might have heard about, so implantation into immune-deficient mice, where they can be grown directly from a patient's tumor in a mouse. Or they can be grown in vitro as cell lines or a sort of more, let's say, next-generation type of cell line called an organoid, where they have a three-dimensional context and probably more closely resemble the tumor as it grows in vivo. Excuse me. And so we've developed a number of these, and we've in the process of characterizing these. And that's kind of, so very briefly, what are some of the things that we are doing and the field will do with them? So one thing that one can do very broadly if one has a large collection of cell lines is to ask, can we define vulnerabilities? Can we define drugs that unexpectedly are very effective when a certain mutation or other feature is present? And many of these studies have been done to date. And I have to just tell you that the published work has largely excluded cholangiocarcinoma because of the lack of models. And so we and others, our lab specifically has developed about 40 models. And so we can now put into this large screen, so this was a large screen that was done a number of years ago, where a number of cancer types, and you can see there's no cholangiocarcinoma here, 112 lung cancers, were analyzed for the response to hundreds of drugs, and now thousands of drugs. And so now we can sort of in a general way ask, can we identify unexpected vulnerabilities? And certainly that's being done by us and others. And the hope is that, again, this is an example of lung cancer, but to do the same thing with cholangiocarcinoma is to, based on an underlying set of features, can we predict therapies that can treat those patients? Now, what about FGFR in particular and the problem of resistance? So how dependent are these tumors on FGFR signaling? So work in the laboratory with these PDX models indicates that, in fact, the tumors are really, really dependent. So if FGFR is inhibited effectively, and that's a key question, is how effectively the drug actually gets there and so on, then you can have a very, very striking response. Now, of course, this is only a short-term experiment here, but this is actually quite durable. This is in a mouse model here that's using a patient-derived tumor from a patient who had an FGFR fusion treated with an FGFR inhibitor. And the tumor really loses the ability to grow, and you can also see it here with a histological test for the amount of cell division. And this brown indicate cells that are actively dividing rapidly. And after they're treated with the FGF receptor inhibitor, actually only for a few days, this completely stops. So this is really telling us that in experimental models and using human tissue, that these tumors are really, really utilizing this FGF pathway to maintain themselves and grow. And so we have to understand better how, in patients, how to take advantage of that dependency and to really exploit it so that responses are truly durable. And secondly, we really need to understand what are the basis for this dependency and how to, again, exploit that. And so, and also to understand how the cancer cells change when they become resistant. So I'll just show this very briefly. This is my last slide here. Complicated slide here, but we can hypothesize for what downstream cellular proteins are engaged by FGF receptor, because it's been studied since the 1980s for its role in how tissues develop and differentiate. And we can see which of these known pathways might be relevant in cholangiocarcinoma and use that to predict ways that we can combine drugs or to predict other resistance mechanisms. So I'll just show you one example. So this is probably the best studied pathway that FGFR signaling uses during development. So it's a cascade of proteins that are turned on. The details don't matter here. And this is what we call an immunoblot or a Western blot. And the dark band here represents a lot. So there's a lot of activation of these proteins here. They're very, very strongly turned on in FGFR mutant cells. But in a patient whose tumor is sensitive, adding the FGF receptor inhibitor completely eradicates growth and completely turns off this whole pathway. And here's another tumor cell derived from another patient who had acquired resistance, and the FGF receptor is, that drug is completely unable to do anything. The signaling stays very high. And we believe that this pathway is probably a major component of how FGFR drives abnormal cells. Finally, so we have models of both sensitivity and resistance. So here is using the FGFR inhibitor drug. IC50 means the sensitivity. So if dots down here mean that the cells, even tiny amounts of this FGFR inhibitor used to treat the cells are enough to completely stop growth. And this has an FGFR fusion, these two cell lines. These ones in black lack any FGFR abnormalities, and they're completely insensitive, as we would expect. They aren't dependent on FGFR. So somebody who has an IDH mutation, you wouldn't have any benefit in being treated with an FGFR inhibitor. They're completely insensitive. However, there are also tumors that have the fusion that gained resistance, and they are completely insensitive in the laboratory. Now, some of them have those secondary mutations that I talked about in FGFR, but many of them, we don't know the basis. And so we're using this attempts to understand the biology downstream to predict, again, ways to both prevent and overcome resistance. I hope that gave a sort of a general overview of how we're trying to subtype cholangiocarcinoma, how we're trying to understand the circuits that support individuals with completely biologically different cholangiocarcinomas. We're trying to develop models to represent the disease as it's manifested throughout the patients in this room. And we're also, we as a community, with very much help from the foundation, is to make more people interested in this area and to understand the incredible opportunities that are there and the importance of the problem. And I think the foundation and the community here has done a great deal of, had a great deal of impact. So the first meetings I went to had a very small number of scientists, and now we were able to invite some of the most famous scientists in America, and we were all working with them. So I do have a lot of optimism for the future of scientific research in this area. And again, I thank you all for your contributions, and I wish everybody well. So thank you.
So
maybe a couple questions and take them together after, since I'm going to do all the clinical trial.
ready? So I was originally diagnosed with one tumor, and they soon did a Foundation 1 result of an NF1. That was my only mutation. I was told I was MSI high and basically, therefore, kicked out of the program. My question then
I'm
is
sorry,
now,
therefore what? I'm sorry?
I'm no longer, that's not an option for me, they said. So now I have 5 tumors. So my question now is, are there different mutations then that can be in these tumors? And then the second part of that, then I'm assuming the only way that I'm going to know that answer is I'd have to have another biopsy.
I'll let Katie comment further on that, but in general, since it's likely that there was a common origin for these tumors, that they arose from a cell that acquired certain mutational events, like the NF1 mutation, probably the likelihood is that they all emerged from NF1 being mutated and giving rise to this abnormal growth. And so it's likely that this sort of truncal mutation would be common. Obviously the most effective therapeutic approach, if there were one available, would be something that targets a common node of these tumors. So I think, Katie, if you want to comment further, but it seems my guess would be that there might be certainly differences between the tumors, but the actual fundamental driver is likely to relate to NF1 function from what you say. I'll also say that just NF1 operates through this MAP kinase pathway. Its basis leads to upregulation of this pathway, and so there are potential clinical hypotheses for how those tumors might be treated. I think Katie might comment.
And just to add,
Could I piggyback on that just for a moment? I'm a nurse and I'm just wondering. I'm with him, and he has a doctor that is willing to treat him with 2 different drugs for that NF1 mutation. Would you consider that a hopeful thing?
certainly to have a targeted therapy, I guess we would want to know more. Maybe we can talk in the break afterwards about which drugs in specific. NF1 is commonly a mutation that goes with microsatellite instability or mismatch repair, which has a lot of mutations that will be follow with it. So it's hard to know if it's a truncal mutation or not in that context. But definitely would be very interested to hear which combination, and perhaps during the break. Maybe should we take 1 or 2 more questions now and then perhaps combine the rest of the questions? I'm going to review a lot of the things that Nabeel talked about from a clinical perspective with clinical trial data. And so, so that we also have, say, a relatively on time, maybe we can do 1 or 2 questions and then switch to see if some of it comes up in the clinical trial section.
Yeah. You said one thing in talking about the BGJ-398 that I found very interesting and was not something I understood before, and that was in the patients who responded well to it, a good percentage of them responded well, and then they started coming back again and having more tumors, and you said that was due to, I think, polyclonal resistance mutations. And then in FGFR2, I guess I was not aware that DNA would continue to change. And so I'm not, so does that mean that each time you go through a cycle with a product, there's a good chance that the DNA that you need to be worried about now is going to be different than the previous test you had, and so you should be getting regular tests of your DNA to see which mutations you now have?
So I'll take that in 2 parts. I think any clinical comments I'll leave to Katie, but that's a very interesting question, and in fact, it hit a sort of conundrum that the field has. So are these mutations pre-existing in very low levels? And most people, I would say that most of the field thinks that these mutations are present already in very, very rare numbers, and when the inhibitor is given, the cells that do not have the mutation are effectively treated by it, and therefore the remaining cells that are rare are the only ones that can survive and are able to propagate. However, this isn't formally proven, and so it is possible that there is some element of new mutations happening. But in terms of monitoring mutations when somebody's on treatment, this is certainly something that's being done as part of many clinical trials, and for FGFR therapeutics, targeted therapeutics, there are drugs that can, in principle, overcome some of these mutations. So it could, I think, eventually be used to guide subsequent therapeutic decisions, but I'll let Katie comment on the actual state of it as a clinical tool rather than a research tool. I can't comment on that, but those are fantastic questions, and part of it actually hits on something that is something that we debate a lot, as to what extent are mutations happening in an ongoing fashion versus pre-existing. So thank you.
Yeah, hi. I was fascinated by your slide showing the near absence of IDH mutations in extrahepatic cholangiocarcinoma. I'm wondering if sort of genotypic discrepancies like this between IHCC and EHCC are really moving us in the direction of thinking of these as less as a sort of geographic subtypes of one disease, more in the direction, as we see in other organ systems, of really almost completely distinct cancer types that happen to share a predisposition for the bile ducts.
Yes, you're distinguishing now intra and extra, or also within a tissue, the different mutational subtypes, was your question?
Are we thinking about intrahepatic and extrahepatic cholangiocarcinoma almost now as distinct diseases?
Yes, I think I'll let Katie comment on that further, but I do have a couple of comments, is that there are some mutations that are quite shared. And so one possibility is that, and this is perhaps more maybe what you weren't getting into, but in the liver there are hepatocytes and there are bile duct cells. And we know in experimental systems that there's kind of an interconversion that can happen. And so conventionally the field thought of hepatocellular carcinoma as being a cancer of the hepatocytes and intrahepatic cholangiocarcinoma as being a cancer of the bile ducts. But it's possible that certain mutations play a role in the identity of hepatocytes. And our hunch, and without a lot of data, is that IDH mutant cholangiocarcinoma might largely be associated with reprogramming of the hepatocytes. And so whereas other cancers, let's say KRAS mutant cholangiocarcinoma, might be derived from the bile ducts, and we have some data indicating that. And KRAS mutations are also frequent in extrahepatic. So it might be kind of a little bit of both, in the sense that a KRAS mutant intra and extrahepatic might have quite similar kind of biology, whereas an IDH might really distinguish the two. But it's a great question.
My question goes along with the gentleman that just spoke here. In terms of the DNA changes after there's been treatment, in particular like with an inhibitor, how often, what would be the protocol that you would recommend for getting additional NGS testing? And my secondary question to that would be, what's the difference between a tumor slide or a tumor specimen versus a blood specimen? Is one give you more data than the other? Is one better in a certain situation than the other?
I'll let Katie comment on it, but I wanted to make sure that I didn't cause any misconceptions, is that when we think of chemotherapy, and people know that a lot of chemotherapies act at the level of damaging DNA, and they take advantage of so cisplatin and drugs like that, and they take advantage of the fact that cancer cells aren't able to repair the DNA. And so the downside of those drugs, potentially, in certain situations, is that they can cause mutations. Whereas, I just wanted to clarify, and I'm sure some people might have thought this, is that it's not that the FGFR inhibitor damages the DNA itself. We have no reason to think that that happens. So I just want to distinguish, these are so-called targeted therapies. We know that they're pretty precise in what they touch in the cell, and they shouldn't touch at all the DNA in any way. I know that's not what you're asking about, but I just wanted to clarify that. But I think to your direct question, I'll let Katie answer.
So I'll touch upon the types of molecular testing and how to test for it in a little bit in my talk as well, which is very clinically focused and clinical trial focused. But right now we don't know how often to sequence tumors, but for certain types of testing, particularly FGFR fusions, tumor tissue is really the gold standard, because cell-free DNA or blood-based tests have a hard time capturing the entire range of the FGFR gene beyond just the coding region. So like Nabeel was giving us a very great primer into, there are lots of parts of a gene that we don't necessarily see in a protein called the introns or the non-coding regions, and that's where some of these fusions or breakpoints can happen, and you need a test that can test all of those potential fusion partners. And the plasma assays haven't yet gotten there for the whole spectrum to date, but we're certainly hopeful that a blood test could do this in the future, and it can pick out some of the resistance mutations, as Nabeel showed, in the actual coding domain, but not necessarily the fusions themselves.